I would love to introduce you to Dr. Alan Stanley. Alan, as many of you may have seen in the Herald article on the weekend, does work as a consultant neurologist here in Hawke's Bay, also has an incredible wealth under his belt around research, clinical development, and just a real passion in this space. So we're so lucky to have him here tonight. He is a board member on motor neuron disease New Zealand. He's also the principal investigator at the Lighthouse 2 trial, which I'm sure many of you are interested in hearing a bit more about tonight. So without further ado, and more babbling from me, thank you very much for listening to me. I would like to open the floor to Alan. And what we're going to do, Alan's going to talk you through his presentation, and then we will have time at the end for some Q&As. So we will get to that. And
right. Okay, well, thanks for coming, everyone. I thought this thrilling tales of the downright unusual, because sometimes advances in medicine and neurology in particular seems a little like science fiction. So I'm going to try and talk you through some of that today. What I'm going to try and do is the sort of brief was inches to milestones, and I really wanted to tell you the evolving story about therapeutics in MND, and rather than sitting up and reading through a list of new drugs that are available, I thought I would try and start at the beginning of this sort of our understanding of the disease and try and intersperse that with some of how it's led to treatments and how that's developed. I'm going to try and touch a little bit on advances in assistive technology as well, but I think I may have bitten off more than I could chew because I've got quite a few slides. So seeing how time goes, we may skip that at the end. So about me, I'm originally from Cape Town. I've been living in Hawke's Bay since 2018. That's a beautiful Table Mountain. And I trained at Groote, which is the hospital where the world's first heart transplant was done, which is a beautiful place to study. And I'm a general neurologist working and living in Hawke's Bay. And my work has really overlapped with MND, predominantly, well, the clinical work, obviously, but mainly through advocacy and trying to motivate for patients and trying to improve networks around clinical trials. So I'm not a primary researcher, I'm more of a advocate and clinical trial enthusiast. So there's an old joke about neurology that we're elephantine in diagnosis and murine on treatment, that we sit around pontificating about these complex networks and then just sort of shrug our shoulders and say, well, there's not much we can do about that. But actually, I think that's a little unfair, and increasingly we're able to do a lot for a number of diseases. We're very good at managing seizures and migraines, and a lot of the immune conditions have really exploded over the last 20 years, advances in stroke, and even advances in our sort of understanding of perceptual disorders and functional neurological problems, which were previously regarded as psychogenic. And I think it's important to start off by saying that we do have care for motor neuron disease, and there's good evidence that MDT care is really extremely valuable, prolongs people's lives and improves people's quality of lives. And I was involved in the team that wrote the best practice recommendations for New Zealand around MND care. And so we do have things that we can do. But what we're talking about today really is disease-modifying treatment. And some nuts are much harder to crack in terms of illnesses. And really degenerative diseases as a group, including things like Alzheimer's, Parkinson's, motor neuron disease, have been quite hard to tackle for a number of reasons. For some, we've developed symptomatic treatments. For some, we just have a diagnosis. And I think that MND certainly is a very tough nut. It seemed very complicated. There are a lot of different strands. There's a lot of clinical variation in how people present. That makes it seem very complex. So just to briefly explain the disease for those of you that aren't familiar with it, it's a degenerative disease of motor neurons. And probably there are environmental and genetic causes for that. It's somewhat unique in neurological conditions in that it affects two parts of the motor system. So the upper motor neuron, which is the brain and spine, which is typically affected by stroke, and people who have illnesses there tend to have sort of stiffness and rigidity. And the lower motor neurons, which are the peripheral nerve and muscles, and people who have problems with that tend to be floppy and loose, and muscles move by themselves. And motor neuron disease is the combination of those, which leads to a fairly distinct combination of clinical signs. So it really is characterized by painless progressive wasting and weakness, generally with no sensory involvement. It has these mixed features, and the weakness affects any skeletal muscle. So it spares heart and smooth muscles, so sphincters aren't involved. And it's obviously a picture of Stephen Hawkins, who's probably the most famous person to live with motor neuron disease, and certainly one of the longest lived. But really, what causes motor neuron disease has still been a bit of a puzzle. So we know about 10% of it is genetic. We know there are these odd, rare cases where a specific toxin induces a very similar syndrome, and the cycad in Guam induced a syndrome called the ALS Parkinson's complex, which looks very similar to motor neuron disease. But for most people, they really don't obviously have themes connected. This is not Harry Potter, that's a young Shostakovich. Charlie Mingus, jazz, a musician, the cartoonist who drew SpongeBob SquarePants, obviously Lou Gehrig, one of the most famous people with motor neuron disease. And Mao Zedong, of course, the Communist Party being what it is, it's a little shrouded if that's actually what he had, but some people do think that he died of motor neuron disease. And what's hard to draw strands from these people's lives, that's long periods of exposure to many, many different things. And how do you nail down what exposure is relevant in terms of the environmental risk? But whatever the cause, it is a devastating illness, and the average mortality is death within 3 to 5 years, with progressive disability in between, which is an arduous journey. There have been some treatments to date. So riluzole was the first treatment licensed for motor neuron disease, and that trial goes back to 1995. It was done with slightly old trial methodology and showed a median mortality benefit of 2 months. So you live 2 months longer on average, which doesn't sound like much. In conjunction with good multidisciplinary care, that is better, but it still leaves a lot to be desired. Newer treatments have come out over the last few years. These really just slow diseases. They work on antioxidant mechanisms, and the ALS-FRS is a functional rating score of disability. You start off with a high number of points, and as you get more disabled, you get fewer and fewer points. And so it really is fairly modest slowing, but that is significant. Of course, the cost is quite significant. So this riluzole costs about $7,000 a year. The newer treatments cost about $150,000 a year. And for just slowing disease, that is quite a cost for health services. Indaverone and Riliviro aren't available in New Zealand, but riluzole is. So what I want to start talking you through is what are some of the cracks in the nuts, and we're going to touch on some genetics, some proteins, and some cell biology. The story, it does get a bit complex in parts. I've tried to keep it simple, so I hope the molecular biologists in the audience will forgive me for simplifying some things. These are the original descriptions of motor neuron disease, ALS, which were from Charcot's original drawings. And he was a really seminal neurologist in that he did very careful clinical descriptions of people and then did autopsies to try and work out what the problem was. And that had huge implications for localization of disease, what functional systems are about. And these are his drawings of the sclerosis, the scarring that happens in the lateral columns, so hence amyotrophic lateral sclerosis, when you turn it into fancy Greek. In going through this paper, I found a brilliant footnote that was apparently written in one of his comments about a colleague, Frederick, who described Frederick's ataxia. And he wrote, such a neglect in matters of nosographic distinctions, especially in a question in itself rather obscure, is at least regrettable and can only maintain the confusion, which I thought was a really fascinating comment. And it's good to see neurologists haven't changed, because we still nitpick the details, and I'll try and use that in future communications. But from there, we learned more about the electrophysiology of the disease, and clinical neurophysiology, where we can test peripheral nerves, started to develop around the early 20s. Denny Brown is another giant in neurology who's a New Zealander, and he described the fasciculation potential, which is one of the key features of motor neuron disease, although we do see it in normal people. And as clinical neurophysiology developed post-World War II, it was only in 1994 that we had the first electrophysiological diagnostic criteria. So you can see there's quite a long lag time in developing these things, and that's undergone several revisions since then. The sort of histopathology of it started off obviously with Charcot, although the staining techniques were pretty rudimentary. Interestingly, this woman is Tatiana Bonina, who described the Bonina bodies, which is one of the hallmarks of ALS pathology, which we didn't know what it was about. She was actually trying to see whether it was infectious and trying to infect mice with cell homogenates. But it was only in the 80s and 2000s that we started to realize what these proteins were and the significance of what was involved. For reference, plaques and tangles are the Alzheimer's pathology, and they were described a bit earlier, and Lewy bodies are the Parkinson's pathology that was also described in about 1912. So the problem, I think, is that you have this disease that has a lot of different clinical manifestations that everyone's approaching from slightly different models. And when you have largely sporadic disease, it's quite hard to know how to develop a model in which you can investigate it. And so one of the ways of tackling this that has proved quite fruitful is, can you tackle an apparently sporadic disease, because 90% of motor neuron disease seems to be sporadic, through a genetic model? And the history of the genetics goes back quite a long way, but wasn't really recognized until the middle of the century. But William Osler, who's another famous physician, described a family in America, which in researching this, I discovered that they've written, a book's been written about them and their journey with one of the main mutations in ALS. And so the description was there in the 1880s, but it wasn't until the mid-50s that a description of that genetics and the possible implications occurred in the literature. And I think that this description of, while hereditary alone may not provide an adequate explanation, a study of the heritability of disease in terms of its physiology and biochemistry might give us the answers. And it really wasn't almost until 50 years later that started to be fully realized in ALS research. And that was really with the first gene, which is the SOD1 gene, which was discovered in 1993. And this allowed us to start using models of disease, and in early work that is the mouse. So not so good for mice, but very good for people, because it's easier to test things in mice. And with techniques in genetic engineering evolving at that time, you could then induce that mutation in a mouse and then study the disease in that mouse and then start to understand the biology, test therapies, and try and bring that back to people. So SOD1 stands for superoxide dismutase, and it's mainly an antioxidant. So many of you are familiar with the sort of idea of antioxidants as being generally healthy and good for you. And that's because mitochondria, which are the engines of our cells, produce superoxides, which are very toxic to proteins and can distort DNA and interfere with cellular functions. And so the main role of SOD1 is to detoxify those. And so in disease, that starts to if you get the protein itself is misfolded and toxic, but you get increased antioxidant damage to the cells. So I think it's worth just stepping back a little just to briefly describe protein. So this is an example of hemoglobin, which is the protein in your blood that carries oxygen. And basically, DNA is translated into these chains of proteins, which then have to be folded to have function. And so you then get the heme ion, which is what binds the oxygen in the middle of this cellular machine, and they bind together. Now what happens when oxygen binds there is that whole structure changes and it hugs the oxygen tighter. And then when it moves through the body to an area where there's less oxygen, it opens up and releases it. So the structure of proteins is quite critical, not only to their function, but also to making sure that those proteins themselves don't do damage, which can happen in disease. So the misfolding itself can then be toxic and can actually knock on and have further effects in the cell. So this diagram is just to briefly show you that what happens with normal folding is that it incorporates various ions to help with that folding, and then you have a functional protein. But in disease, these proteins start to misfold, they don't sit together well, they start to clump in awkward ways. That gunge collects in the cell and interferes with its function. And they also break down and start to mutate and knock on and influence other proteins. And so that cascade of disease is what was gradually unpicked. And the interesting thing that came out of this work is that mutations aren't the only things that can damage proteins and lead to misfolding. So lots of environmental and cellular processes can disrupt the proteins. And with good models, you can then explore, how does this happen? What are the cellular processes? What are the biomarkers that we can use to research this in people? And you can start to test therapies. So this isn't quite how the story went, but basically this is one of the drugs that was tested in the SOD1 model, the other lines of research, and it's a copper-containing substance. And you saw in the previous slide that copper is one of the metals that helps the protein fold appropriately. It penetrates the blood-brain barrier well. And in mouse models, it was shown to prolong survival, improve motor function, and help with mitochondrial function. And there was an extremely positive phase 1 trial in 2019, and they were a little naughty when they published the result, because they said that it led to a 70% reduction in motor neuron disease progression, but really that trial wasn't designed to show that at all. And so that headline-grabbing soundbite led to a lot of stress. People wanted to access disease well ahead of the time that it was actually ready for clinical use. There has subsequently been a phase 2 trial, which was actually finished in 2021. And frustratingly, the company hasn't yet released the results, which I presume means it's been negative. But these are the kinds of things that can develop once you have a model. You can test diseases and try them out. And I think it's critical for discovery. So like when they discovered the microscope, suddenly there were these rather forbidding, strange creatures that were all around us. And actually now we have these rather forbidding, strange diagrams of cellular processes that are all around us. But it allows you to iterate and understand and develop things more quickly. So just to briefly touch again on some basic science, because I'm going to launch into genetic therapies. You'll be familiar that DNA is the encoding material of our bodies, and everything about us is encapsulated in these strands. It lives in every cell in the body, and that's double-stranded and has base pairs that translate. Every 3 base pairs get translated into a protein, which then form the chains. Now the way that happens is that you get the DNA gets opened up, translated into single-stranded RNA that then is incorporated into the cellular machinery and builds proteins, which then fold, as I showed you earlier. What's really fascinating is that allows us a way to have genetic therapy. And so this is a technology called antisense oligonucleotides. And what happens is that if you have a messenger RNA that's leading to a faulty protein, you can introduce a dummy opposing sequence that can block that translation, and so that the RNA can be read off the gene, but then because there are these antisense oligonucleotides floating around, they bind that RNA and don't allow it to be made into the faulty protein, thus preventing disease. And as of last year, we have the first genetic therapy for motor neuron disease. So this is only for the specific SOD1 mutation, so it doesn't apply to all people with motor neuron disease. And it was registered with the FDA in about the middle of last year. And what the initial trial showed was that the mutant SOD protein is dramatically reduced. They measured a biomarker called neurofilament light, which is released by destroyed neurons. And certainly those biomarkers dramatically improve. They designed the trial slightly awkwardly. So they designed clinical to measure statistical significance of outcomes at 6 months, and actually that's probably too early. So the clinical outcomes reported in that study were less robust and didn't meet the significance. But in the 12-month follow-up study, there's been quite significant divergence. And also the earlier you treat with it, the more function you seem to save. And there have been some very encouraging improvements in grip strength, and that people certainly seem to stabilize over time. And these have now been used in trials for a number of years. And some people have had really long-term benefit from it. Biogen does have a compassionate access program for this now. So they're funding for free 300 patients worldwide. People who are skeptical of pharmaceutical companies say, well, nothing is compassionate about pharmaceutical industry, and they're just trying to corner market share. But nevertheless, it does allow people who have this mutation to potentially access treatment. And there is at least one patient in New Zealand outside of a trial setting receiving this on that basis. So it's amazing that most of this work came from discovering the genetic mutation. Of course, there were other improvements in cell biology and immunohistochemistry and all sorts of other staining going on as well. The problem is that SOD1 isn't representative of most MND. So this is, it accounts for about 10% of familial cases and only about 1% of sporadic MND. So there are still cases if you apparently don't have a family history and you do genetic testing, you do find mutations. But luckily, we discovered a lot more since that initial description. And in 2006, we realized that the main protein involved in gumming up the cells in motor neuron disease is something called TDP43. And not only is it that involved in motor neuron disease, but also in frontotemporal dementia. And clinically, we do see an overlap between those two diseases, that about 20 or 30% of people with motor neuron disease will develop frontotemporal dementia. Some people develop more mild cognitive changes. And the progress of genetics discovery really has ramped up over the last couple of decades, as you're well aware, and we now know a huge number of genes involved in motor neuron disease. This is an older slide, but I like it because it does show those that are the most significant. C9orf is a more recently discovered gene, which is present in a number of sporadic cases as well. And you can see the purple and green that implicated in proteome homeostasis, so keeping your proteins well regulated at appropriate levels is in green, and purple is about RNA binding proteins, all of that sort of DNA transcription mechanics. And this is newer work showing that, so if the middle circle is the familial ALS cases, more than half of those we now have known mutations for, and there must be mutations in the others that are just harder to find. But very excitingly, a big chunk of people with sporadic ALS with no family history turn out to have genetic mutations as well, meaning that disease treatments and mechanisms that we see in researching these genetic familial conditions have implications definitely for these people, but also the mechanisms that we uncover will probably influence treatment for other people who don't have a known mutation yet. So what does TDP43 case do? So it's present in 97% of MND cases, both familial and sporadic, and in frontotemporal dementia. And it's predominantly a protein that lives in the nucleus and helps to regulate some of those transcription mechanisms. What happens is that in disease, it starts to get stuck outside the cell and misfold. Now, some of that is from mutations in various sites, but some of that is from environmental stresses. And this is a common point of convergence, that even sporadic cases have TDP43. And so something triggers it in them, even though we don't know what that is yet. And so these misfolded proteins then have a very similar behavior to the SOD1 protein, that they clump up and they start to gum up the machinery. And what we've realized is that some of that is about the clearance of these proteins. So some of it's about the production, but some of these mutations are in the clearance mechanisms that help to process. Your body obviously is self-healing and knows that this protein is abnormal and should be cleared up. But if that system doesn't work properly, then that can also lead to disease and contribute to it. So this is what's a medical talk without a forbiddingly large table that you can't read, but it's just to point out that TDP-specific therapies are in trials. So these are things that have come to trials. Some of them are phase 1, some of them are moving through to phase 2, and really they're focusing on this biology of trying to increase the clearance, trying to prevent that misfolding, and some of them are trying to tackle it through multiple mechanisms. What's interesting is that some of these are repurposed medicines. So colchicine down here is a gout medication. It's very cheap. It's been around for decades and decades. And if that works, that allows people to instantly take an off-the-shelf treatment. And there are a number of strands of these medications that are being repurposed from other conditions that have plausible mechanisms and can be tested. So what about C9orf? This is the most common mutation that we know about to date, and it's rather complex. So it does seem to be involved in DNA repair and cell transport and autophagy, which is that clearing up of gummed proteins. But what's tricky about it is that it's not only a gain of function. So if you have a mutant protein that's creating too many disordered proteins, it's also if you have too little of it, it also seems to lead to disease. And that makes it a little tricky to tackle because you can't just block it off at the source, because then you lead to disease as well. And so, but nevertheless, there are some trials. So there have been 3 antisense oligonucleotide trials. 2 have unfortunately failed. 1 is still going on. Some of these others are looking at mechanisms by which the proteins are produced and shepherded. And then metformin, which is another drug, which I'm sure many of you are familiar with for diabetes, is important in blocking the system that helps to make these proteins. And so that's another really appealing example of a potential for a medication that's widely available and cheap to be repurposed for treating complex diseases. Metformin's having a bit of a moment in all sorts of different things. A lot of people feel it's broadly anti-inflammatory and everyone should be on it, and it's a panacea at the moment. So, but evidence probably doesn't quite back that up just yet, but it is a very interesting molecule. So to change tack a little bit into cell therapy, whenever you talk about degenerative diseases, people talk about, well, what about stem cells? Stem cells must be the answer, because these are the building blocks of life. They're where we all come from, and surely this is the answer. And they're different, just to touch on the terminology, pluripotent stem cells are from embryos, and they have the potential to turn into anything. As you differentiate down the cell line, the cells get more and more stuck into their adult conformation and do their jobs. You do still have multipotent stem cells, mainly in bone marrow. And so that's where bone marrow harvesting and stem cell harvesting for bone marrow transplants come from. The problem is trying to tell those stem cells what they should turn into and where they should go. So many stem cell therapies that are available in the community, people will go to clinics of varying repute and get a bag of something delivered into their veins. Now, even if those are stem cells, you can't necessarily tell them where to go and what to become. So I imagine no one would want to put stem cells into fat, but fat's a pretty straightforward tissue. If you put a stem cell there, maybe it would turn into fat. But trying to get it in to be a nerve, to go to the right system in the brain, connect to the right things, is obviously a much bigger task. There are some mechanisms that stem cells home to injured tissues, and there is some plausibility to it. But by and large, it's really not selective enough, certainly not for treating neurological disease. There is an interesting therapy that's in development. It's been submitted to the FDA and has been initially declined, although doubtless there'll be some further research. Now, this is taking stem cells from a person and putting them through a process that doesn't turn the stem cell back into nerves. It teaches it to make stuff that's good for your nerves. So it encourages neurotrophic factors and helps to deliver sort of antioxidants directly from your own cells. And so it's a very interesting therapy. There is some research for it and some clinical trials, but the evidence is still a little mixed whether that really works. But it's an interesting way of trying stem cells to deliver these neurotrophic factors. So I don't know, our children are mid-teenagers now, but when they were born, there were certainly a lot of companies around trying to encourage us to deep freeze the umbilical cord in case we needed stem cells later for us and them, and we better save them now because it'll all be gone. I don't know about you, but we didn't. But don't worry, because luckily we can now make them, and I think this is almost the most science fiction thing I've heard in the last few years. So what you can do now is you can make stem cells, not from bone marrow, but from a skin biopsy. So you can take a little piece of skin, you can put it into a lab, you can culture the fibroblasts from the skin, so you get a cell culture. Then you can teach it to turn back into a stem cell. So you give it some cytokines, you turn it back into stem cell, and it turns back into pluripotent stem cells, the original embryonic ones that can be turned into anything. And then subsequently, we've learned how to turn it into virtually any tissue you imagine. So you can now take a cell culture from a person with a disease with a certain set of genetic mutations and environmental exposures. You can take a skin biopsy and have stem cells of their disease, and you can turn it into the tissue of interest and study that in a dish. And I just think that's so amazing. It's really unbelievable. It's not quite at the level where we can then turn that back into a neuron and connect it into the brain. But certainly in terms of a disease in a dish model and studying disease, and a lot of therapies are now being tested in these models. And that's much better. It's much better for mice. It's much better for us because it's easy to cure things in mice, but humans are more complicated and we're not mice. And so there are numerous therapies that were successful in mice and have failed in humans. And of course, if you have a cell culture that you can just grow for a month or two, test a drug, doesn't work, no bother, you just bring out the next tray and test the new drug. So the potential for rapidly testing drugs is hugely expanded. And it's more relevant because you're using specific patient-derived tissues. Obviously, there are other possibilities for cell therapies, and some of these are, hopefully one day we will actually be able to tell them where to go and do the right job. They can modulate the immune system, they can neurotrophic factors, as we showed you, and then microglia are part of the immune cells of the brain, and they can be taught to be anti-inflammatory as well. And so there's some interesting work around that. The other thing that I'm sure everyone's aware of is a CRISPR-Cas9 gene editing system. So this has been around for more than a decade now, but this actually allows you to edit DNA. So you can use this system to find a specific segment of DNA and cut it out and replace it. Of course, the problem is that you don't want to tell it to only do that bit of DNA. And so a lot of the work that's going on around this is about targeting it specifically, trying to make sure that it doesn't grow a cancer nearby by some unintended off-target effect. But there are, in other diseases, evidence or successful trials. So the first CRISPR-based therapy was registered also last year for sickle cell anemia. And there's a trial currently going on for another protein condition, amyloidosis, where an abnormal protein is made in the liver and goes out and clogs up body tissues, and that's currently in development. With respect to motor neuron disease, that's been used predominantly in cell culture models because we're still learning about how to target it and how to use it, but it's definitely going to be something to keep an eye on over the next while. So I just wanted to briefly mention the trial that I've been involved in. So my role is actually kind of largely administrative. I help to fundraise for it, and the Neurological Foundation and the Motor Neuron Disease New Zealand generously sponsored this project. And this is another repurposing trial looking at a different mechanism of disease. So only about 1% of our genes code for tissue. So that's what the sort of coding region of the DNA. The rest of it is regulatory material and other stuff. And about 8 or 10% of that is viral DNA. And so there are these things called retroviruses, of which HIV is the sort of most modern version. That part of how they work is they insert their material into our genome and then use our body's own machinery to read that off and generate whatever's of use to the virus or make more virus. And that's how they cause disease, and that's how HIV causes disease. But these have been going on throughout our evolution. And some of these very old ones, HERV-K is the one that's particularly of interest in ALS, were inserted a really long time ago. And there's evidence in motor neuron disease that those are reactivated and there's viral proteins around. And interestingly, the proteins that help to transcribe that are targeted by HIV therapy. And so we're using an antiretroviral that was developed to treat HIV, combination antiretroviral, to try and see if we can modulate this response and downregulate that toxic protein. So we had funding for 20 patients in New Zealand, which doesn't sound like much, but it's one of the first sort of broadly accessible clinical trial available. We've recruited 19. There have been all sorts of staffing issues. There's one patient still to be recruited in Christchurch. And we're applying for more funding as part of a collaboration with Australian colleagues. So hopefully we will be able to get more patients on it. At the time that we put this together, we didn't have enough patients in Hawke's Bay. And so that's why I'm not actually running a site, but I'm the sort of national coordinating investigator for it. So really, I think that's this sort of medication development strategies. That's the fascinating story, is that we've gone from having no models of disease and no way to effectively test medications other than giving them to patients, and it's very slow. We were sort of off target. We didn't really know what the mechanisms were, to discovering some of these genes, being able to generate mouse models, and now induced pluripotent stem cell models, disease in a dish. We're using our technology to model these cellular processes in computers, which I haven't touched on, but that's another way of ramping through, trying structures of proteins, which protein and which chemical might interact with that, and you can generate therapeutic targets on that basis. We've changed how we do trials. So from the original trial, trying riluzole, where it was just a standard single patient gets a single placebo, we now have platform trials where we run 5 or 6 trials at the same time. You use the same placebo group, so you only have to give that to one of the 5 groups, and you test 5 or 6 different therapies concurrently, and they're much more agile and adaptive and have been used successfully in cancer. We're looking at different biomarkers. We've got different ways of measuring disease, and one of the other interesting things about tofersen, the SOD1 therapy, is that was one of the first therapies to be approved by the FDA on the basis of biomarker results rather than clinical effectiveness. So it's just a tricky thing. Is a biomarker really the effect? But there's increasing evidence that that is the case and that those are valid therapeutic markers. So I think that the thing that we're left with is what causes motor neuron disease. And for 90% of people, it comes out of the blue, and it's a disease that seems to strike out of nowhere. What we understand is that there are certainly environmental exposures. There's a lot of debate about exactly what those are, but some of these do seem to be things like smoking and chemical exposures, military deployment, head injuries, exercise, excessive exercise, that is, probably with concussions, and sometimes just the mutations that you acquire just by going through life. We all have mutations in our genes all the time, and that's like cancer. You pick up a mutation and it leads to a cancer, and degenerative disease and motor neuron disease in particular is probably part of that. But in modeling, we know that you probably need about 6 steps to reach disease. Now, if you are born with absolutely no disposition, you have to get all of those 6 steps. So you have to have a lot of damage over time to trigger a disease. If you're born with a very strong mutation like SOD1, you may only need a very minor environmental trigger to tip you over into getting that disease. And most of those diseases show what's called age-related penetrance. So you inherit the disease, it's a dominant condition, but you don't develop disease until later in life. And so C9orf, for example, is some evidence that if you live to 85 as a man, you have a 100% chance of getting disease. But at age 50 or 60, that's only 50 or 60%. And so it does show that you'd need those sort of environmental exposures to trigger the condition. Probably for most sporadic cases, a lot of those are not single genes that we've discovered so far. It's part of the general variation in our genome, some of which will be significant and some of which won't be. And there are fascinating ways of trying to study that, because you have to compare a person's DNA with a database and find that variation. And that's much easier if you can find related people. There's very clever research going on in Australia at the moment where they call it cryptic interrelatedness. So they look at databases of patients who've donated genetic material and find out that actually some of them are distantly related. And that allows you to do much more powerful genetic analysis. So they don't know they're related, but turns out they are when you analyze their genes. So this is, again, an older slide, but I think it illustrates some of the heterogeneity of the illness. So you have different clinical pictures, some of which have mostly upper motor neuron and some of which have almost all lower motor neuron. You have these overlap of dementia and motor neuron disease. You have different anatomical regions of onset, some variation in the actual pathology, but also the spectrum of genetic variation from very high penetrant dominant mutations to some of these that are milder but just portray risk, and then in probably sporadic people, just a complex polygenic, similar to many chronic diseases like diabetes. It tends to run in families, but we don't, there's not many single gene causes of diabetes. And this is just a sort of optimistic slide from putting currently recruiting trials for ALS into the clinicaltrials.gov, which is a trials database, and there are 137 trials worldwide currently recruiting patients. And not all of those are clinical. Some of them are biobanks and natural history studies, but many of them are therapeutic. And I think our current challenge is to try and access this. And what I think we'd like to do as a country is to try and get a network of people together that we can try and attract these trials. Now, some of that is community involvement, and you need people living with the disease to do slightly boring research like prevalence studies and incidence studies and how much does it cost to live with the disease. And people don't necessarily want to be in that kind of research because it's not a therapy, but that helps to set the groundwork for being able to motivate why this is a good place to come and do research. Because you have to sell us as a country and an infrastructure that we can successfully do research and we have the appropriate knowledge about the disease in our country. And part of that's how much do we have? What age does it present? Where is it? What are our risk factors? And so being involved in that kind of research, and the M&D registry is a really important portal into that, where patients can register, describe the disease, the onset, the diagnosis, and collect a standard battery of statistics. And some of that doesn't generate information for that person. It's a voluntary thing, and that is quite hard for people to see that the long-term benefits of that registry are really valuable. And that's part of what allowed us to get this trial, is that we had some of that knowledge already, and we're planning more of that sort of scaffolding research, at the same time trying to generate these networks to try and attract clinical research. So therapeutics summary, the pace of development's dramatically increased. Our understanding of genetics has really helped us understand the disease and the processes involved. And because of that understanding, we understand the disease more, we can try more treatments, we can bring things to trial much more quickly. If you look at the pace of change in some of those slides, from SOD1 being described in 1993 to a trial in the sort of mid-2010s to C9orf within practically a decade, having 3 different antisense oligonucleotides being trialed for it and other molecules. So I just wanted to say, thank you to my colleagues, because I'm really an advocate and a sort of trial administrator, whereas the people in the Lighthouse team there are the people actually enrolling patients and doing the research on the ground. Dr. Emma Scotter is a hugely influential basic science researcher. She's a passionate patient advocate and does really brilliant basic science research. Dr. Claire Riley is a person living with motor neuron disease who's been heavily involved with the Motor Neuron Disease Association and community for many years. And she was the dynamic spur that attracted a lot of people into the community. And of course, everyone else who's contributed to M&D research, Sir Richard Fall, who's put together the Brain Bank, Chris Shaw, who's recently returned to New Zealand from London, where he's been doing a lot of this basic research, and he's going to be bringing very exciting trials to New Zealand, and many others. I had a pause there to see how we were doing for time, because I have some stuff on brain-computer interfaces, but I think it's already 7:20, so maybe we'll save that for another day. Thanks for your time.
Hello, is this one on? Fantastic. Alan, thank you so much. That was utterly fascinating. We are going to open the floor now to questions. Can I ask you please just to raise your hand and to speak into the microphone? I'll go one side, Susie will go the other, just because this is being recorded, and so to capture the questions, it would be great to get you to speak into the mic. So, who would like to open the floor with any questions? No?
They don't have to be too technical.
Thanks, that was a very interesting talk. I was struck by one of the slides where you talked about treatment for gout being repurchased. Can you just give us an idea of what causes a researcher to suddenly look at something like gout treatment as a possible, have other benefits?
I think one of the main tools there are these in silico, so in computer modeling. So now that you know what a protein looks like, you can run it through databases of other chemical structures to see what chemical structures might interact with that protein or that substance of interest, and that generates a number of interesting therapeutic targets. Some of it is an understanding of sort of shared pathways of disease, so that if you know that a particular inflammatory pathway turns out to be relevant in leading on to the mutant protein or disease of interest, you can then look at medications that are known to have an impact on that pathway. So there are a few different ways that it can come through. The other thing with induced pluripotent cells is that you can do high throughput assays, so you can test the number of batteries of substances and find which ones look interesting, and then progress those further.
I was just wondering if these blood cleaning tablets that help to clean blood vessels, whether that has any research done with regard to motor neurone disease or other neurological diseases?
Yeah, blood cleaning is kind of an interesting term. I don't think there's anything specific that I could point to. I mean, the closest would probably be the sort of broadly active antioxidant type things. So like the copper that treats, antioxidant mechanisms. I don't know of any sort of blood filtering or blood cleaning products, I'm afraid. But I'm definitely not up to date with everything. So.
Hello, I'm a member of the general public and came here tonight just out of general interest. The only thing I really understood in any sort of depth is that you talked about autophagy.
Yes.
And there's been a huge interest and following for intermittent fasting, which is supposed to clear some of that gunk, clear up the... do you think that's a fact?
Well, I must say I don't know of a specific MND related trial, but certainly in longevity research, there are people who are very interested in those mechanisms. And the idea basically is that our systems should be more stressed than they are. And when we get too comfortable, stuff starts to go wrong. And so that the idea is that you need a certain amount of physiological stress to kind of keep things processing and keep things clearing out. And certainly there does seem to be some evidence for that in some of the that sort of longevity research. How exactly it would apply in motor neuron disease is not 100% certain. There is a neurologist in Hamilton who's a huge fan of the ketogenic diet, and he has several. He's done a trial on Parkinson's, which looked very interesting, and he recommends it for patients and has produced a number of case reports which look very interesting. But I don't think we don't have enough information at the moment. But it certainly is a potentially interesting mechanism.
Lovely, thank you very much. Do you mind me asking what the chap's name is, who's the ketogenic chap?
Matthew Phillips.
Phillips?
Yeah.
And what's his first name?
Matthew.
Matt.
Yeah.
Thank you very much.
Alan, is AI helpful in what you're doing at this point?
Yeah, so I think I'm not sure, but these massive computing power certainly is, and undoubtedly there will be AI techniques for doing that. I think one of the things about AI that I'm always a bit suspicious about is you don't know how it got to the answer, which always makes me just suspicious of what it's come up with and how much of it it's made up along the way. I mean, you've got plenty of stories of people who've tried to get some paper or references. My colleague did a fascinating journal club. He's a respiratory physician about asking AI to generate a piece of information, and he realized that some of the references were just made up. And then he sort of challenged ChatGPT and said, "Why did you just make this up?" And he went, "Oh, I'm sorry, I'm not supposed to do this." It's like so it's kind of fascinating dynamic, and so it is a bit of a black box. But yeah, there certainly will be. It's a massively powerful processing tool, and I'm sure there will be applications for it.
Any other questions?
Are you familiar with David Sinclair? His work on diseases and...
I think so, if we're talking about the same Davidson.
Yeah, I think he's mainly interested in finding the root cause of disease. Like you mentioned that motor neuron disease could be amongst people with Alzheimer's and dementia, so a whole family tree. And his focus was on what's the root underlying cause. And I was just wondering if you had any thoughts on, instead of attacking it from a drug from on top, if you like, to identify, like, is it really senescence or cells that have gone astray and become mutants that are causing these diseases? Is there something underlying that we can attack?
Yeah, I think that's, there are interesting shared mechanisms across neurodegenerative diseases, and the vast majority of the things that are really troublesome to us as a community are proteinopathies. So there are various accumulations of proteins, and so it's not implausible that there might be a root cause, but I think I've tried to show that there's quite a lot of cellular mechanisms involved, and these are, because they're such complex systems, it's very hard to pin down just one trigger. And probably it seems more plausible that there are going to be a number of potential exposures that can set off that sort of cascade. So I'm not sure if we'll ever reach a single one. The sort of easy diseases like chimney sweeps getting bladder cancer, leads to quite a tight environmental exposure relationship. These diseases are much more complex because they come later in life and there's so many more exposures. So hopefully I think there will be similar mechanisms, but I'm not sure if there'll just be one root cause.
Yeah, is motor neuron disease a first world problem, or is it throughout the world?
No, it does seem to be universal throughout the world. So I trained in South Africa, and we certainly saw a lot of motor neuron disease as well. It seems reasonably stable through populations. There are some variations, and obviously a few years ago, we have the dubious honor of having the sort of highest incidence. That is a slightly estimated incidence, but we're not sure exactly why that is. But it's certainly seen, it's not a disease of only of sort of Western diet, like heart disease or obesity or those sorts of things. Yeah, so it is sadly a relatively universal problem. Yeah. The certain mutations obviously vary between populations, and interestingly, I was just reading recently that C9orf, I think they suspect there's a founder effect in Finland, and it might be the Viking expansion that helped to spread that particular mutation. So another thing to blame on the Vikings.
I think we have time for one last question.
Yes, the toxin thing. We had a son that we lost through motor neurone, and he worked in one particular port and in New Zealand, and there were 9 people that got motor neurone, and there was a banned subject. The substance has now been banned, but it was methyl bromide that was put in the hulls of the boats.
Yeah.
And is there a link with that? Has there been a link with that?
Well, it's one of the things that's been mooted as a possible cause, but it doesn't seem quite as straightforward as it's just exposure to that leads to it. Certainly it's, it definitely has been explored. And it is interesting, we do see these occasional clusters where you have people on neighboring farms or in the same town or working in the same port, where there's suddenly an apparent cluster. And it's that's the kind of thing we need to study, and we need the sort of that community infrastructure and ways of identifying people and being able to sort of pool that shared experience and see what their exposures were and what's going on with them to try and help. So it is certainly on the list of things that's a possibility, but I don't think it's been definitively shown. But these things are always quite complex. I'm sorry for your experience.
Well, Alan, thank you so much. Please join me, ladies and gentlemen, in thanking Alan for a wonderful talk this evening. And thank you again to all of you. Thank you so much for joining us this evening. If you are new to the foundation, we would really love to keep in contact. I know Susie and I have been encouraging people to grab one of our magazines on the way out. Please do keep in touch if you're interested. And some of the work that Alan's talked about tonight, plus some of the other research that is going on, has all been, as I mentioned, funded through the community, through people just like yourselves. So thank you so much for showing up tonight, for your passion for neuro research and for supporting ourselves in this community. I'm guarantee everybody in this room has had their own experience with a neurological condition, either themselves or a loved one. So thank you for sharing with us this evening. We are going to stay for a little while and have some tea and coffee. There's some light snacks at the back. Please do grab some material on your way out. We would dearly love to keep in contact with you all. We hope to come back to New Plymouth. Well, I'm sorry, we were in New Plymouth last week. I hope to go back there too. But also we hope to come. Yeah, we do hope to be back here shortly. So it'd be great to hear from you if you're interested in keeping in touch about events as well. So please join me once. Let's say thank you once more, and then please enjoy a cup of tea.